NURS 2533, Process of Human Disease 1. This is Unit 3, Part 3. So it's again chapters 8, 9, 10, 12, 40, 45, and 52. So today we're going to be talking in Part 3 about infections. So infections really have to do with invasion by microorganisms. And there's a couple of basic types of microorganisms, basic classes of microorganisms that cause infections. Uh, Three of them are here. It's bacteria, viruses, fungi. Also, we could add parasites and prions, but we won't go there quite yet. Protozoans can also cause infections, but the three main ones are bacteria, uh, viruses, and fungi. So, of these types of microorganisms, most of them are not pathogenic. They don't infect humans. They don't cause anything with humans. Uh, some of them are part of our normal flora and they're symbiotic. They actually are beneficial. They produce vitamins for us. They do all kinds of good things for us. Um, the vast majority of them don't even care if we exist. So there are more types of bacteria in the world than any other organism alive. A few of them are pathogenic, a few of them are symbiotic, helpful, and 99.9% of them don't affect people at all, one way or the other. The same is true of viruses, fungi. Our environment is full of fungi, um, but not necessarily going to be a problem with us. So the ones that we're worried about are the pathogens. Pathogens mean that they cause disease, generates or the genesis of patho disease. Um, Another word that we use a lot is microbe to refer to these pathogens. Um, Now we'll start out with bacteria because bacteria are the kind of the most common or a very common cause of infection. It's the one we seem to know the most about, and so yeah, we're going to just start there. So bacteria are cellular organisms. They are made of cells. However, they are prokaryotes. They're not eukaryotes like we are. Uh, the cells are different in that there's no nucleus. They have DNA, they have chromatin, they have um, other organelles in them, but they don't have membrane-bound organelles. So they don't have endoplasmic reticulum and they don't have Golgi apparatus and things like that. They don't have mitochondria. So uh, they they're different when you look at them. They also reproduce differently. They reproduce by binary fission. They get to a certain size and they just squeeze off, and some things go one side and some things go to the other. They don't require another cell to reproduce. There's no, for the most part, there's no sexual reproduction with them. Most bacteria have really complex cell wall structure. Um, they are the cellular wall is more than a cell membrane, and there's some complexity to it, and that's what gives the bacteria its shape. We call the shape and size morphology, and there's all kinds of different morphologies in bacteria. If we go back to this slide, we can see that here's some of the morphology. If the bacteria are little spheres, they are they're considered cocci or cocci. So if there's two spheres together, they're diplococcus. If there's a grouping of the cocci, they're staphylococcus. If they're in a chain, a stripe, they're streptococcus. So the cells are little spheres. In if it's a bacilli, they tend to be more elongated. So the cocobacilli are kind of just ovals. Uh, they can be rods. They can be fusiform. Right? They can line up like a fence. That's a palisade. Another form that they take is spirochetes. Um, Syphilis, Lyme disease, uh, etc., are spirochetes. They are kind of corkscrew shaped. So the bacilli, rod shaped, spirochetes uh, are spiral, and the cocci are spheres, diplo, strepto, staphylococci. So all bacteria have this cell wall, and this is what protects it, gives them a specific shape. And the cell wall has some differences. And for our purposes, I mean, to a bacteriologist, there's a lot more differences than just this. But from a medical perspective, it's useful to kind of put the cell wall into one of two categories, gram positive and gram negative. Now, gram is a type of stain, so gram stain, you put in the culture of bacteria, and if the cell wall picks up the stain and stains purple, it will be a gram positive bacteria, and if it doesn't, it will be a gram negative bacteria. And how these cell walls are will really have um, a predictability as to what the clinical effects will be, but it also, you need to know this to choose what kind of antimicrobial therapy that you're going to use. Gram negative bacteria are different and will respond to different antibiotics than gram positive bacteria. So it's really very useful to know the difference. Um, well, let's actually go back to the differences between gram negative and gram positive. So a gram negative bacteria, um, it stains kind of a pink in color. Not that that matters to you, but it does. And the cell wall is made of lipopolysaccharides. They're kind of sugars and fats together. Um, and and these lipopolysaccharides will will actually um, induce cytokines. They cytokines like tumor necrosis factor, which is TNF. Um, they will cause inflammation. They can cause fever. Um, they're responsible for septic shock. More than the gram positive ones are. They they change more readily, they mutate more readily, and they become antibiotic resistant. The bacteria that are becoming antibiotic resistant tend to be the gram negative bacteria. The, they're a common cause of infection. Uh, the and they cause a lot of symptomatology. Now, to contrast that, the gram positive are purple when they stain, and they cause fever as well. And you can't clinically distinguish whether the infection is a gram positive or a gram negative. A lot of the same factors. Um, but they seem to um, take hold and infect people that are immunocompromised. Uh, a lot of these gram positive are just kind of normally in the environment, like Staph aureus and things on the skin, and they take advantage of an immunocompromised person, um, and that's when they get infected. So that's the fundamental difference. Now, bacteria, one of the things that they do is they release toxins, and toxins are any chemical that causes damage. So, uh, endo or exotoxins are secreted from the cell to the outside of the cell, so into the host. Um, so they they are made, but they are not. They're secreted, and so they work outside the this the cell. Gram positive and gram negative are capable of secreting exotoxins. And what does the exotoxin do? Well, it varies. It depends on the exotoxin. It depends on what the action is. Most of them are destroyed very easily just with some heat. This is kind of what the whole point of pasteurization is. But it really doesn't take a lot of heat to destroy most, except for Staphylococcus enterotoxin. And enterotoxin means that it affects the intestines. Um, it, Fairly low heat, like 60 degrees will do it. So some examples of exotoxins that are important to humans are things like tetanus and Botox, botulism toxin, uh, diphtheria. You don't actually get sick because of the infection of the bacteria. You get sick because of these chemicals that the bacteria secrete, the toxins that are being secreted by these bacteria. Now, endotoxins are slightly different. Endotoxins are present in that cell wall. And so usually it's the gram-negative bacteria. And the endotoxins don't get released until there's lysis of that bacteria, until that bacteria is killed. And then what happens is these endotoxins, these chemicals that are part of the cell wall that get released into the interstitium, they induce inflammation. They make interleukin 1 release. And you get all the clinical manifestations of inflammation. So you can have the vasoactive properties. You get shock, blood pressure drops can happen. It causes fever systemically. You can end up with sepsis from these endotoxins, and these happen when the bacteria is killed and it's released at that point. So now viruses are um, are different. Viruses, there's a question as to whether these are even life forms. Um, that's pretty much been accepted that they are now, but nevertheless, for the longest time, because they're almost too simplistic. Now, it says that they're obligate intercellular. It should be intracellular parasites. They 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 need to live inside the cell. Um, they they can't uh, reproduce outside of cells. They have no organelles to them. Viruses are a very simple form, and all they really are is this protein coat called a capsid, kind of like a capsule, and some sort of DNA or RNA. So it's nucleic acids inside this protein coat. Now, some of them are encapsulated, some of them are enveloped uh, in a lipid bilayer, in kind of a cell membrane. We'll talk about where that comes from in a couple of minutes. But they really are pretty simple. All it really takes is the capsid, the protein capsid, and RNA or DNA. Um, Now, it can be either or. Some viruses actually have an enzyme or make an enzyme that will allow the RNA that makes the virus to convert to DNA. These ideas are important for treatment. Um, the HIV virus, the human immunovirus, is one of these things. So it's an RNA virus, but it hijacks the DNA synthesis of the cell. So it actually has to use this reverse transcriptionase to convert the RNA to DNA in order to reproduce. And so one of the current drugs that they use for people with HIV infection is a reverse transcriptionase inhibitor that prevent this from happening, and it slows down the reproduction of the virus. Um, Now, the virus uh, really, it to reproduce has to go in and take over the transcription and translation of protein synthesis of the cell and use that to make the protein capsids and to reproduce the nucleic acids. Because they're so simple, it doesn't take very much change, and the proteins can change a little bit. So they are very quickly mutating. Also, generations don't take very long. So mutation can happen very quickly in on the surface uh, envelope, the surface proteins. Um, and that makes it very difficult for uh, the immune system to recognize and have memory of these viruses. So oftentimes, because of this quick mutation, every new one that you encounter is a primary response rather than a secondary response. It's the thing that really distinguishes the influenza virus, why we have different strains of viruses all the time, and that you can get the flu over and over again. Fundamentally, the virus hasn't changed. What's happened is the surface proteins, the antigens, have mutated and changed, and it's like your immune system is seeing it for the first time. So, what happens here? The virus ends up in your body somehow in the interstitium, and then it attaches to the receptors on a cell. And this is why certain viruses are affect certain tissues and organs more than other tissues and organs, because it takes advantage of the receptors that each cell has to its own function. So, for instance, the HIV goes in through the CD4, and so then affects the helper T cell, infects the helper T cell. Other viruses use other receptors to gain entry into the host. So they attach to the receptor, and then they inject their genetic material, not necessarily the protein coat. The capsid usually stays on the outside. What ends up happening is the genetic material goes inside, and then that that genetic material goes and is reproduced in transcription and produces new proteins by translating And so it takes over that protein synthesis of the cell. And so the new viruses get assembled within that cytoplasm of the cell, and the cell just fills up with virus, all new virus. Then one of a couple of things happens. Either the cell just dies, undergoes apoptosis, and when that happens, the viruses are released. Or sometimes the viruses bud. They just they push out through the cell membrane, and when they do that, they actually take a bit of that lipid bilayer with them and form their envelope. So the envelope viruses come from this. And that's obviously hard on the cell because the cell is losing cell membrane when this happens, and it will happen until the cell just dies. Or the third way that they get out is through exocytosis, uh, where they really hijack the cell, and the cell just undergoes normal exocytosis, and in those vesicles, it is the virus is being released. So a lot of the like for instance, viruses that affect neurons could could use the neurotransmitter exocytosis route, um, things like that. So somehow the virus gets in via receptor, takes over the protein synthesis, makes new viruses, and then they leave either by killing the cell, budding out, or by being through exocytosis. Now, the next group of organisms we're going to talk about is the fungi. Now, fungi are different than the other two because they are eukaryotic organisms. So that means that their cells actually have a nucleus, and they have endoplasmic reticulum, they have organelles, uh, and they are organisms. Now, they're everywhere. We have fungi everywhere in the environment, uh, and most of them don't do anything. A few of them will cause disease. They're pathogenic, uh, but even then, they tend to either be local uh, or uh, or somehow not very virulent. A lot of them end up growing up on the skin, so they're primary infections of the skin. So these are fungal infections. So this is be things like ringworm and athlete's foot. Those sorts of infections just grow on the skin. Some of them in the mucous membranes, things like thrush in the mouth, and that kind of thing. Uh, they happen a lot of times when somebody is immunosuppressed. Now, it is possible to get a systemic fungal infection, but those people that get that would have to be really immunosuppressed. So you'll see opportunistic fungal infections in people with AIDS, with full-blown uh, acquired immunodeficiency. Uh, you'll see thrush and other fungal infections in people that are on immunosuppressive drugs for transplants and for things like that. It's a problem with people that are undergoing cancer treatments, chemotherapy. Uh, Fungi take advantage of. For the most part, healthy people, fungi aren't a huge problem. Um, and but it's still entirely possible. Okay, so those are the three major ones. We can also have protozoans, which are also eukaryotic organisms that, so things like Giardia, um, amoeboidosis, beaver fever uh, can be caused. They tend to be gastrointestinal because we tend to ingest protozoans. Um, Again, not a broad category, not like bacterial or viral infections. So, a couple of definitions to do with infection. Sporadic is when the infection happens in a single individual. So, somebody gets tetanus. That would be uh, a sporadic thing. Endemic means that there is constantly trading back and forth, that it's within the population, endemic, in population. So it just goes around and round. Now, epidemics are when there's more than usual, that and it spreads to a larger area. Pandemic is when it covers the entire world. So sporadic is the least, and pandemic is the most. So I like this concept here, this chain of transmission of microorganisms, and this is bacteria, this is viruses, anything. In order for uh, infections to occur, we need all of these things to be in place. And by looking at it this way, we can also look at ways of preventing infection. Um, Now, the first thing that's necessary is a source of the bacteria, a reservoir, as it were. Now, the reservoir could be other humans. A lot of diseases spread human to human, things like HIV. The reservoir is a human that's infected. That that other human could be ill with that disease, or they could maybe be a carrier of that disease, like typhoid. There are people that don't have clinical manifestations of typhoid, but they become carriers of typhoid. They're the reservoir. A lot of times, reservoirs are insects. Zika virus is mosquitoes. Could be animals. Could be the soil. Things like tetanus are in the soil. So somehow and somewhere the is a host, a reservoir of the pathogen. And then that pathogen somehow has to get out of the reservoir. So that's the portal of exit. So it might be the nasal mucosa in the case of influenza. It could be oral mucosa. It could be uh, urinogenital. So it could be the mucosa of the genitals. It could be, you know, whatever. Somehow that pathogen has to have a way out. And then it's got to get a way to be transmitted to the next person. So it could be through insect bite, it could be through semen, through coughing or nasal droplets, things like that. It could be airborne or it could be waterborne. An interesting thing I read last year was that uh, it looks like the diseases that are waterborne are more virulent. They cause more symptoms than the ones that are airborne. Um, and it really is in the pathogen's best interest. If a, the pathogen makes you too sick, then it's going to block its mode of transmission. But if it's waterborne, really sick people still pee and they drink and they're bathed, and so the mode of transmission doesn't rely on the person being up and ambulatory. Other things, the airborne transmission really requires the ambulation of the other person. Um, so then the transmission has to get to the new infect person that's going to become infected. It needs a way to get in. So skin punctures, that's how tetanus can get in. Skin abrasions through various mucosa, nasal mucosa, oral mucosa, um, very common portals of entry. And then the victim has to be susceptible to the problem, to the microorganism. So they have to be either immunocompromised, or it might be a novel pathogen that so they're unimmunized, they have no memory, they've never encountered it before. Uh, they might be malnourished. It might be all of the above. And so when we look at infections, and especially from an epidemiological point of view, we have to look at all of these different things, and we break the infection in one of these parts of the chain. So so if the vector is a mosquito, and so that's where the reservoir is, then eradicating mosquitoes is a good idea, or eradicating mosquito habitat like standing water uh, is is good. If it's a waterborne, then sewage treatment uh, is probably a good idea. Like those are the sorts of things. You destroy the reservoir if you can. Um, if it's rabies, then removing rabid animals uh, would be the way of blocking it, of destroying the reservoir. Now, blocking the portal of exit is a good idea. So isolation basically is a real good thing for some infectious diseases. You don't want to spread the disease, so you isolate the person that is the reservoir, and you make it so that there's no real portal of exit. Masking, using gloves, using condoms, all of these things are physically blocking the portal of exit. You can block the mode of transmission by using sterile technique, hand washing, thorough cooking of food, uh, you know, pasteurization, things like that is block the mode of transmission. Blocking the portal of entry is a lot like the portal of exit, so mask, gloves, condoms, and basically the difference is the giver and the taker. Uh, it's the same sort of thing, just who is doing the blocking. Now, probably one of the better ways of dealing with this is to reduce the victim's susceptibility. So, good hygiene practices, optimal rest, nutrition, vaccination programs are all looking at doing this. It, it, the whole idea is to break this chain of infection. Okay, so there are barriers that protect us. We talked about them in AMP2. We've talked about them in part one of this unit. Um, we talked about the mechanical barriers and biochemical barriers in the mechanical barriers. So the fact that we have mucus, we have intact skin, we have stomach acid, we have various secretions on the skin, and prostate and testicular secretions. We have cilia uh, that move the mucus around and move it to places where any that has a microorganism and that'll get destroyed. Most of the secretions in our body are acidic and they have lysozymes in it. There's resident flora in some of the openings in the vagina, in the large intestine, in the guts. Um, they and oftentimes people are susceptible to infection when those resident flora are removed. Um, there's a there's a good school of thought that says that vaginal yeast infections have to do with decreased vaginal secretions or excessive cleanliness or removal of flora, sometimes too much antibiotic use, those sorts of things. Um, a, a big problem in the hospitals right now is C. difficile, Clostridium difficile, and that happens because of excessive antibiotic therapy actually killing the resident flora in the gut. And then the C. difficile colonizes. Basically, there's a vacancy that the bacteria takes advantage of. So by having a healthy gut actually prevents a lot of infections. There are some research being played around with uh, basically fecal transplants, where you take a healthy person's poo, kind of mix it with water, and then give an enema to a sick person, and you try and replace their flora with the healthy person's flora. Um, and it seems to be showing some really promising results. It sounds a little distasteful, but it works. Now, this Venn diagram really is looking at the interaction between the microbe, the environment, and the host. And a lot of things overlap. So acute illness, fever, stress, broken down mechanical barriers, lack of phagocytes, chronic illness, all are host things that predispose to this infection. Poor nutrition, poor hygiene, a buildup of toxins, age are all host related things. But there's an overlap here. Nutrition and hygiene kind of go with sanitation, water quality, living conditions, weather, seasons, air quality. A lot of that kind of environmental aspects will predispose to infection. Now, the microbes really are, um, it's like, what kind of encapsulation do they have? Do they live outside of the host for very long? Adhesions. So adhesions are important in, say, infections of the urinary tract. Urinary tract infections, you would pee away the pathogens unless they somehow adhere. Um, or their flagella. Are they transmitted through mosquito bites, arthropods? Those sorts of things all have an overlapping effect. So a an old immunocompromised person with poor nutrition and poor hygiene, living in crowded conditions in mosquito uh, environments where the right type of mosquito makes them susceptible to malaria, which uh, you know is the microbe. So those things all come into play, and each one of these things are things to address to try and prevent infection. Now, to switch gears entirely, we'll look at the course of an infection, the normal course. Um, there, so infection happens, the pathogen starts to colonize the host, and the immune response is either slow or unable to eliminate that pathogen. So at first, the pathogen is in you, it's reproducing, there isn't enough of it to actually cause signs and symptoms yet, And uh, we call this period the incubation period. Um, there's there's no intensity, and the numbers of the microorganism are very low. Then, as the numbers start to increase, then so do the signs and symptoms, but the signs and symptoms are vague in general. And we call this the prodromal period. And this is the time when people are saying, I think I'm coming down with something. I just don't feel myself. Then the symptoms, especially the specific symptoms of that disease, start to manifest and they increase. And this is the acute illness stage. Uh, and at this acute illness stage, it gets worse and worse and worse until the immune response is able to start to beat it. And as the immune response starts to eliminate the pathogens, then the illness goes into decline and you start feeling a little bit better. And then as the 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 whole thing ends in a convalescence period where the immune system is just cleaning the remainder up. And this is the normal course of an infection. This slide looks at kind of the same idea. The infection starts here at time 0. There's an incubation period. Now, this is the line of, of kind of the number of pathogens that where you get symptoms that manifest clinically. You get clinical manifestations. We call that the clinical threshold. Now, if the number doesn't reach that clinical threshold, you become kind of a reservoir of this disease, but you don't get sick yourself. And so we call that a subclinical disease. Now, this prodromal stage kind of lasts from one side of the clinical threshold to the other side of the clinical threshold. It's not, you're not really sick. And then in the acute phase of this, the pathogen is winning. The pathogen is ahead of the immune system and reproducing, and you get more and more symptoms. You get more and more ill. Now, at this point, this is called the critical threshold. If you cross the critical threshold, you can't recover, and death is the result of that. Now, at some point before the critical threshold, the immune system starts to be able to get control and gets the upper hand and starts to be removing the pathogens faster than they can reproduce. Now, this is the period of decline and moving into convalescence. And the immune system is getting rid of more and more of the pathogens. The pathogens are having harder and harder time to reproduce, and so they're going away. You still have some pathogen in once you cross the clinical threshold and you're still convalescing. Resolution happens when there is no more infection. Now, sometimes the the immune system can't completely eradicate it, but can keep it kind of at bay. And this is when you get chronic disease. So this would be things like these are the incurable infections, things like herpes or HIV infections. All that we can do, we can't get rid of it. But we can keep it close to that clinical threshold, if not below the clinical threshold. Now, this is a list of various bacteria and where they commonly infect. So by no means do I want you to memorize these. You'll be taking a microbiology, a micropathology course when you get to York, and so you'll be going over them then. But you'll see that some of them end up in a number of places, like Staph aureus ends up in the eyes. Neisseria gonorrhea ends up in the eyes and the urethra. Uh, so staph will end up in various other places, like on the skin, right? Clostridium is in the liver, but it's also in the intestines. So strep pneumoniae is in the lungs, right? But strep pneumoniae is a big cause of meningitis. So uh, these are the areas that these bacteria are fond of going after. So now, how do you know when somebody has an infection? So you look at the signs. Now, here's an interesting thing. The signs of a local infection in one area, are really a lot of the same signs as inflammation. But remember, infection causes inflammation, but not all inflammation is infection. So you have to be very careful. So at some place that's infected will be swollen, but not all swelling indicates infection. Erythema, which is redness, the same. Pain and tenderness, the same. Lymphadenopathy is really swollen lymph nodes. That's a sign of infection, and a lot of times it's like, oh, do I have an ear infection? Check my cervical lymph nodes. Yeah, they're palpable, therefore it's a good chance. Purulent exudate is a pretty good sign that you've got an infection locally. So if if it starts to ooze pus, then chances are good that it's an infection, probably bacterial infection. Now, when you go to systemic infections, again, we've talked about a lot of these things. Fever, leukocytosis, so increased white blood cell count, elevated ESRs, and C-reactive proteins are blood tests. Fever, weakness, anorexia, fatigue, headache, joint pain. These are all flu-like symptoms, you'll probably notice. So that's really signs of systemic infection, which are a lot of them are the same as the signs of systemic inflammation. Now, there are various tests that we use, especially for bacterial infections. Nothing beats culture and sensitivity tests. So this is where you take a sample, you culture it, you grow the type of bacteria, you look at it and identify it, and you test it with antibiotics to see what it's sensitive to and what it's resistant to, so that you can develop a treatment regime based on this test. Blood tests that indicate would be things like leukocytosis, cytosis, an increase in white blood cells. If it's a viral infection, you actually may get leukopenia. If it's an acute bacterial infection, the number of polys will be up, so you get more neutrophils. If it's chronic, you'll have more monocytes, more lymphocytes. We look at the C-reactive proteins and the ESR. We talked about that. They indicate inflammation, and because infection causes inflammation, that's indicative that there could be things. Now, the other thing we look at is imaging. So the reason that the dentist takes an x-ray is to look for abscesses to see if there's an infection below the root of the tooth. So all in all, infection is a common thing. It has a normal course. It's probably the thing that you will see the most in clinical practice. So the next unit will be stress and disease. It's a much shorter unit than this unit, but we will do that for the next section.